If you really want to understand trees, you have to understand how they are built and how they function. Every pruning decision, every diagnosis, every management recommendation an arborist makes is ultimately rooted in tree biology. When you look at a tree, you're not just seeing bark, leaves, and branches. You're seeing a highly organized living system made up of specialized tissues, hormones, transport pathways, and growth structures. And for the BCMA exam, understanding these biological systems is essential. In this podcast, we're going to explore the internal biology of trees. We'll look at how trees grow, how they move water and energy, how they respond to damage, and how they regulate growth through chemical signals. By the end of this lesson, you should be able to picture what's happening inside the tree when you see growth, stress, or decline in the field. Tree biology can be understood through two major lenses. First is anatomy, which is the physical structure of the tree. Second is physiology, which describes how those structures function and interact. We'll explore the tissues that form the vascular system, the way trees transport water and carbohydrates, and the role of plant hormones in directing growth. We'll also examine how trees respond to injury through processes like compartmentalization and how they regulate seasonal changes such as dormancy and senescence. These concepts appear constantly on the BCMA exam, but more importantly, they show up every day in arboriculture practice. Let's start with the fundamental organization of a tree. A tree is composed of different types of tissue, each specialized for particular functions. These tissues form the tree's vascular system, which transports water, minerals, and sugars throughout the plant. Two major components of vascular tissue are xylem and phloem. Xylem primarily transports water and dissolved minerals from the roots upward. The phloem transports photosynthate, which are sugars produced during photosynthesis, from areas of production to areas that need energy. Those areas of production are called sources. Mature leaves performing photosynthesis act as sources. Areas that use or store sugars, such as growing buds, developing fruit, or roots, act as sinks. Inside the phloem, sugar transport occurs through specialized conducting cells called sieve tube elements in angiosperm trees and sieve cells in gymnosperms. Water movement through the xylem occurs through structures called vessels in tracheids. A common BCMA exam concept is recognizing the difference between angiosperm trees and gymnosperm trees. Angiosperms are flowering plants that typically contain vessels in their xylem. Gymnosperms, like pines and spruces, generally rely on tracheids for water movement. Within the trunk, xylem tissue forms the wood of the tree. New xylem produced each year becomes part of the secondary xylem, while the conducting outer layers are called sapwood. Over time, older xylem becomes heartwood, which no longer conducts water but provides structural support. When you cut a cross-section of a trunk, you often see growth rings. These rings reflect seasonal changes in wood production during secondary growth. In some species, the pores appear evenly distributed, which is known as diffuse porous wood. In others, large early-season vessels form a distinct band, creating ring porous wood. Growth rings also help arborists identify reaction wood, which forms when trees adjust to mechanical stress. In gymnosperms, reaction wood appears as compression wood. In angiosperms, it forms as tension wood. Now let's talk about how trees actually grow. Tree growth begins in regions of actively dividing cells called meristems. The apical meristem is located at the tips of shoots and roots. It produces primary growth, which increases the length of stems and roots. The apical bud, sometimes called the terminal bud, protects the apical meristem at the tip of a shoot. Nearby, you'll find axillary buds or lateral buds located at nodes along the stem. The region between nodes is called the internode. The apical bud exerts control over the growth of lateral buds through a process known as apical control. This process is largely regulated by the plant hormone auxin. Auxin moves downward from the apical meristem and suppresses the development of nearby buds. When that terminal bud is removed through pruning or damage, suppressed buds may activate and produce new shoots. These shoots may develop from dormant buds or even from adventitious buds that form in unusual locations. The resulting growth often appears as epicormic shoots along the trunk or branches. For the BCMA exam, understanding how pruning influences apical control is extremely important. Trees also grow in diameter through secondary growth. This occurs in the vascular cambium, a thin layer of dividing cells located between the xylem and phloem. The vascular cambium produces secondary xylem toward the inside and secondary phloem toward the outside. This radial expansion is responsible for increasing trunk diameter year after year. Another important cambium layer is the cork cambium, also called Phellogen. It produces phloem to the outside and phelloderm to the inside. Together, these tissues form the periderm, which replaces the original epidermis as protective bark tissue in mature stems. You may also notice small pores in bark called lenticels. These structures allow gas exchange through the otherwise protective outer bark. Inside the tree, transport occurs through two major pathways. The Apoplast pathway moves water and solutes through cell walls and intercellular spaces. The Symplast pathway moves substances directly through the cytoplasm of living cells, connected by plasmodesmata. Water enters roots through the process of absorption, driven largely by osmosis. Osmosis is the movement of water across a semi-permeable membrane from areas of lower solute concentration to higher solute concentration. Fine absorbing roots and root hairs dramatically increase the surface area available for water uptake. At the root tip, a protective structure called the root cap shields the delicate meristem as the root grows through soil. Surrounding the root system is the Rhizosphere, a dynamic zone where roots interact with soil microbes. Among the most important of these microbes are Mycorrhizae, which form symbiotic relationships with tree roots and significantly enhance nutrient uptake. Once water enters the tree, it moves upward through the xylem, largely due to transpiration. Transpiration is the evaporation of water from leaves through tiny openings called stomata. Each stoma is controlled by two specialized guard cells that regulate opening and closing. This water loss creates a tension that pulls water upward through the xylem column. Another critical biological process is photosynthesis. Within leaf cells, chloroplasts capture light energy and use it to convert carbon dioxide and water into carbohydrates. Pigments within the chloroplast play important roles in light absorption. Chlorophyll is the dominant green pigment. Carotenoid pigments produce yellow and orange colors. Anthocyanin pigments contribute red and purple coloration. These pigments often become visible in autumn as chlorophyll breaks down during leaf senescence. Leaf drop occurs through a process called abscission. At the base of the leaf petiole, a specialized abscission zone forms. Cells in this region weaken until the leaf detaches. Environmental signals such as photoperiod are detected through a pigment system called phytochrome, which helps trees measure day length and prepare for seasonal dormancy. Plant hormones, also known as plant growth regulators, coordinate many of these processes. Auxin influences apical control and directional growth known as phototropism, which is a form of tropism where plants grow toward light. Cytokinins promote cell division and bud development. Gibberellins influence stem elongation and seed germination. Ethylene regulates processes like fruit ripening and leaf abscission. Together, these plant hormones form a complex regulatory system that guides plant development. Now, let's discuss wood structure and defense. Wood consists largely of cellulose fibers reinforced with lignin. Through lignification, cells become rigid and strong. Within wood tissue, parenchyma cells play important roles in storage and metabolic activity. Ray cells extend radially across the wood and help with radial transport of nutrients. The center of the stem contains the pith, which was part of the earliest growth of the shoot. When trees are wounded, they cannot heal damaged tissue the way animals do. Instead, they isolate damage through a process known as compartmentalization. This process limits the spread of decay organisms within the wood. In some species, structures called tyloses may form inside vessels, helping block pathogen movement. As the tree produces new tissue around wounds, it forms woundwood, which gradually grows over exposed areas. Field-based arborist examples. Imagine you're inspecting a mature oak after storm damage. You notice a large pruning wound from years ago. Around the edges of that wound, you see ridges of woundwood slowly closing the exposed area. Inside the trunk, the tree has likely initiated compartmentalization to limit decay spread. Now imagine a tree growing at an angle on a hillside. The tree responds by producing reaction wood. In hardwoods, it appears as tension wood, while in conifers, it forms compression wood. This structural adjustment helps the tree maintain stability against gravity. Diagnostic thinking for arborists. Understanding these biological systems helps arborists interpret what they see in the field. When you observe epicormic shoots along a trunk, it may indicate stress or the loss of apical control. When you see poor leaf expansion, you might consider limitations in essential elements needed for photosynthesis and metabolism. If leaves wilt during drought, the issue may involve transpiration exceeding the tree's ability to maintain turgor pressure in cells. And when decay progresses through wood, knowing how compartmentalization works helps arborists evaluate structural risk. Exam insight moments. For the BCMA exam, you will often be asked to distinguish between structures or processes that sound similar. For example, understanding the difference between xylem and phloem transport, or between primary growth and secondary growth. Another common exam topic involves recognizing how plant hormones like auxin influence apical control and bud development. These biological relationships are foundational to advanced arboriculture. Concept reinforcement. So let's tie these ideas together. Trees are built from specialized tissues, organized into a vascular system that moves water, nutrients, and carbohydrates. Growth originates in meristems, with apical meristems driving primary growth, and the vascular cambium producing secondary growth. Water moves upward through xylem via transpiration, while sugars move through phloem from sources to sinks. Plant hormones regulate development, while seasonal signals trigger dormancy, senescence, and abscission. And when injury occurs, trees defend themselves through compartmentalization and the production of wound wood. Summary: tree biology provides the foundation for everything arborists do. When you understand how tissues, hormones, and transport systems work together, you begin to see trees not just as structures, but as dynamic living systems responding to their environment. In the next chapter, we'll examine biotic disorders, the insects, pathogens, and other living organisms that affect tree health. Because once you understand how a tree works internally, you're far better equipped to manage it successfully in the real world.
Thanks for listening to the Arborist podcast. Let's keep building your arborist knowledge one domain at a time. If you found this useful, please like and subscribe.